The Effect of Vitamin Type on Planarian Regeneration Rates by Anu Daga
and Shreeja Tangutur. Planarians are a type of flatworm that are uniquely known for their regenerative capabilities. They possess a unique type of cell known as neoblasts, which are specialized blastema cells. Blastema cells are cells that are capable of regeneration. These neoblasts can divide rapidly, allowing planarians to regenerate quickly. Planarian structures are similar to human structures since they both have a central nervous structure, brain, eyes, intestines, epidermis, and digestive systems. Although human regenerative capacities are limited compared to planarian regenerative capacities, they both have similar embryonic stem cells in their tissues.
Our research question is, which vitamins can accelerate the regeneration of tissues in planaria? With the answer to this question, we can apply this knowledge to human tissue cells, since they have similar structures. People with severely damaged tissues can be helped. We hypothesized that if the planaria receive vitamin C, then the regeneration rate will be faster because vitamin C allows cells to divide rapidly. Dr. O.R. Pagan is a prominent researcher in the planaria field, and he has done much research on the neuroscience and structure of planaria. He wrote the book, The Neuroscience of Planaria, and is currently giving lectures about the usefulness of studying them. He is doing research on the internal structures of planaria.
For this experiment, we chose 3 different vitamins to test. We chose vitamin A, which is known for creating soft tissue, mucous membranes, and aids in reproduction properties. The second vitamin that we chose was vitamin C, which helps the body absorb iron, maintain vital tissues, and contribute to stem cell development by allowing stem cells to divide rapidly. The third vitamin that we chose was vitamin D3, which also helps with the creation of soft tissue, calcium absorption, and immune health. Vitamin D3 also controls the homeostasis of neoblasts, helping the organism grow stronger. Each of these vitamins has the potential to heal tissues and speed up neoblasts, which is why we decided to test these specific vitamins.
For our procedure, we first set up 12 Petri dishes in 3 rows of 4. We labeled the rows Trial 1, Trial 2, and Trial 3, and labeled the columns with control, vitamin A, vitamin C, and vitamin D3. For each Petri dish, we added 30 milliliters of spring water with a graduated cylinder. We added 1 drop of the corresponding vitamin for a certain row, and we mixed the water so there would be an even distribution of vitamin water. We filled an extra Petri dish with spring water and ice to immobilize the planaria, then transfer the planaria back into the corresponding Petri dish.
After, by using a surgical scalpel, we cut the planarian into 4 millimeter pieces. By using the digital caliper, we measured the planaria to make sure each of the pieces were equal in length. We transferred the pieces of planaria to a Petri dish and repeated these steps for the rest of the planarian. Once a week, we fed the planaria 0.5 grams of boiled egg yolk. We made sure to measure each piece of planaria each 5 days and record the raw length. Lastly, they will be kept in a container in the fridge for 48 hours before we dispose of them in a bag. This is what our setup looked like at the beginning of the experiment versus the end of the experiment. Some planaria tend to shrivel up, which is why some look bigger than others.
Our data table depicts the raw length of the planaria over 15 days. We have our trials and our control group with the specific vitamin that aided them with regeneration. By looking at the results, we can see that the planaria in vitamin D3 grew to the largest length. The planaria in vitamin D3 grew the most, followed by vitamin C, then vitamin A, and the control grew the least.
Vitamin D3 is crucial for tissue homeostasis and the creation of tissue cells, which is why it was so effective. The results show that all 3 vitamins tested on the planaria show high regeneration rates than compared to the control. The graph on the right shows a bar graph, and as we were looking at the average growth for each vitamin. This graph also supports our data as vitamin D3 had the largest average daily growth. One problem we encountered was that the planaria shriveled up and expanded constantly when in water. This made it difficult to dissect them, and by doing research, we found that ice water immobilizes planaria. Putting the planaria in ice water allowed us to get accurate measurements and helped us cut when dissecting.
While our hypothesis was invalid, we learned that vitamin D created the most profound impact. Since planaria neoblasts perform the same housekeeping functions as tissue cells in humans, and the structure is alike, we can apply these same concepts that we learned from planarian regeneration to humans. There are many tissue-related injuries that can cause tissue damage or loss. Inflammatory arthritis is a group of conditions that affect your immune system. However, instead of attacking your immune system directly, they attack your tissue cells. Using the results of this experiment, we can conclude that vitamin D3 is the most effective vitamin that can counter the effects of inflammatory arthritis and other tissue-damaging diseases such as sprains. Finding out which vitamin aids in neoblast and stem cell creation, we can apply this to humans and find which vitamin can help restore tissue cells and aid in fighting arthritis.
For further research into this subject, we wanted to contact Dr. O.R. Pagan, who is a prominent researcher in the planaria field. We emailed him in hopes of learning about neoblast and planaria in depth. We asked him what allows neoblasts to regenerate in planaria so quickly. He told us that no one really knows why planaria are so adept at regeneration, and also that different parts of the planaria develop at different rates, and most of the time we should look for the eyes. This information was very useful, and we are currently working with them to figure out the potency of vitamin D and experiment the proper dosage needed to effectively help and replace damaged tissue cells, and how we can administer the vitamin in the correct places of the body.